Very good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and here we are to discuss the most advanced questions from Cell the Unit of Life for our NEET 2026 practice. Let's start here, and the first question is assertion reason type of question, ma'am. So in this assertion reason type of question, the assertion is about non-membrane bound organelle, that is ribosome. Ribosome is prokaryotic cell and it has 70S type of ribosome. Yes, that is true, right? Ribosomes in prokaryotic cell are what type? 70S type, with larger subunit 50S and smaller subunit will be 30S. In reason it is clearly mentioned that in the S in ribosome stands for Swedberg unit and the measure of the sedimentation rate during the centrifugation. So in reason it is clearly explaining about the 70S type, okay, wherein S is standing in ribosome, S stands for Swedberg unit. That is clearly explained in the reason. So here assertion is true, reason is true, and reason is the correct explanation of assertion. Okay, so here we have to go with which option, ma? Option A is the right answer. Next, which of the following is or are not the functions of Golgi apparatus? What is the primary function of Golgi apparatus? Packaging or formation of glycolipids and glycoproteins and synthesis of lysosomes. This is the function of Golgi bodies or Golgi apparatus. So here if you see in the options, formation of glycoproteins, that is a function of Golgi bodies. Sorting and packing of proteins, yes, that is again the function of Golgi bodies. Lipid breakdown doesn't happen in Golgi bodies. Breakdown of fatty acids or oxidation of fatty acids will happen in mitochondria, not in Golgi bodies. Formation of lysosomes again occurs in Golgi bodies. So which is not the function over here, ma? Option C will be the right answer. Next question. Match the following cell organelles with their current function. So we have to match the cell organelles with their function. Golgi apparatus, ultimately this is involved in what? Packaging and modification of proteins and lipids into glycoproteins and glycolipids. Smooth endoplasmic reticulum is purely involved in the synthesis of lipids. Ribosomes is involved in the synthesis of proteins. Lysosomes will help in intracellular digestion. Lysosomes helps in what, ma? Intracellular digestion. So here which option goes good with this question? A is 2 and B will be 1, C is 3 and D will be 4. Option A will be the right answer over here. Next question you look into it, which of the following statements is incorrect they are asking. So we have to identify the incorrect statement. Mitochondria contain single circular DNA and 70S ribosome. Yes, that is correct. Both mitochondria and chloroplast are semi-autonomous or self-duplicating organelles because they have their own DNA. Okay, they have circular double-stranded DNA and 70S ribosomes. This statement is the right answer. Okay. Plant cells have plasmodesmata for cytoplasmic connections, correct? Plasmodesmatal connections in between two cytoplasm of the cells will traverse across the cell walls and the plasmodesmata is made up of desmotubulin proteins. So yes, this is also true. Next, animal cells will have large central vacuoles for storage. This is a wrong statement because large central vacuole is identified only in a plant cell. In plant cell, vacuole will occupy how much space, ma? 90% of the volume of the entire cell is occupied by plant vacuole, not by the animal vacuole. And in option D, if you see, prokaryotic cells will divide by what? Binary fission. So here which statement is incorrect? Option C will be the incorrect statement. Next question you look into, again assertion reason type of question based on centrosomes. Centrosomes are found in animal cells but not in most of the plant cells. Centrosomes, yes, they are identified only in the animal cells, not in the plant cells. In reason it is explaining like plant cells will perform mitosis without the formation of spindle fibers. Wrong. In plant cells, spindle apparatus formation will take place, but then it is not from centrosomes, it is from microtubular organizing complex. Okay, it is from microtubular organizing complex. This complex will be synthesizing spindle apparatus, or this will aid in the formation of spindle apparatus for the cell division to occur in a plant cell. Clear, right ma? So here assertion is true and reason will be false. So what option we have to go with? A is true and R will be false. Okay, next. Look at this question. Which of the following statements are true, they are asking. Okay, pili will help in conjugation. Conjugation, which is one type of sexual recombination that happens in between F plus strain and F minus strain. What is F plus strain, Vita? F plus strain where F factor is present in a plasmid DNA. Okay, F factor is present. So this is F plus strain. So this F factor is transmitted to the plasmid DNA of F minus strain. Okay, this is mediated by what tube? Conjugation tube, which is also called as sex pili or true pili. Clear, right ma? This is also called as sex pili or true pili. So pili help bacteria in conjugation. Correct statement. Next, plant cells do not have lysosomes. That is wrong. Plant cells will also have lysosomes. All the type of lysosomes will be present. Primary, secondary, autophagosomes, residual bodies, all the type of lysosomes will be present even in the plant cell. Next, ATS ribosomes are found in the cytoplasm of eukaryotic cells. True, ATS ribosomes are found in eukaryotic cell cytoplasm, whereas 70S ribosome is identified in eukaryotic cells, mitochondria and chloroplast. Remember that. Next, fourth statement if you have to look into, prokaryotic cells will have double membrane bound organelles. That is wrong, because prokaryotic cells will not have membrane bound organelles itself. So here, which is wrong statement? Option 4. What are the correct combination of statements here ma? 1, 2 and 3. Okay, so option A will be the right answer over here. Next question you look into, again assertion reason type of question. Assertion: in amoeba, contractile vacuoles will help in osmoregulation. When we are discussing about vacuoles, we have come across three types of vacuoles: contractile vacuoles, gas vacuoles and food vacuoles. Okay, of which contractile vacuoles in amoeba will help in osmoregulation. That is correct. In reason it is explaining like vacuoles in protists function primarily for digestion. Wrong. Never vacuole is mediating the function of digestion, because digestion is not carried out inside the vacuole, right? Okay, so reason will be wrong. Vacuole will never aid in the process of digestion. So reason is wrong and assertion is true. In that case, which option we have to go through ma? Option C will be the right answer. Next, match the following type of question. Here if you have to see two columns. Mitochondria, which is a double membrane bound organelle which has its own DNA. We have already discussed mitochondria and chloroplast. They are self-duplicating or semi-autonomous organelles. Next, lysosomes, they are active at acidic pH and they are rich in hydrolytic enzymes which aid in the process of, which will help in digestion of molecules. Okay, those primary lysosomes attaching to the digestive particles will turn to what? Secondary lysosomes, which are also called as heterophagolysosomes. So yes, lysosomes contain hydrolytic enzymes which are active at acidic pH. Next, Golgi apparatus, they will form what? Vesicles for secretion. These vesicles can be what? Glycoproteins, glycolipids or lysosomes. Next, rough endoplasmic reticulum. Rough endoplasmic reticulum are studded with what? Ribosomes. That is why RER is involved in protein synthesis. So here all combinations will go with A1, B2, C3 and D4. In that case, option A will be the right answer. Next question, assertion reason type of question again. The inner membrane of mitochondria is folded into cristae. Yes, once let us recall the structure of mitochondria. Mitochondria, we have an outer mitochondrial membrane and inner mitochondrial membrane which is folded into cristae. Okay, inner mitochondrial membrane which is folded into cristae. The space in between the outer mitochondrial membrane and inner mitochondrial membrane is called as intermembrane space, and inside you will be having mitochondrial matrix. Clear, right ma? Now you see the inner membrane of mitochondria is folded into cristae. This is a right statement in assertion. In reason, if you have to look into, cristae will provide increased surface area for aerobic respiration. Reason is true. So here in reason, the correct explanation is there about cristae, right? In that case, what option we have to choose? A and R are true, and R is the correct explanation of assertion. So option A will be the right answer. Next question, which of the following are true for chloroplast? So the right statement is asked with respect to chloroplast. They are double membrane bound organelles. Yes, that is right. They are double membrane bound organelles. They have 80 S ribosomes. No, chloroplast will be having how much ma? 70 S ribosomes. Okay. Thylakoids are arranged in grana. True, thylakoids are arranged in grana. They possess circular double stranded DNA. This is also true. So what are the true statements over here ma? A, C and D. These are the right statements with respect to this question. Next you look into assertion in reason type of question. Ribosomes are called protein factories of the cell. Correct, right? Ribosomes are called as protein factories of the cell. In reason, they are involved in DNA replication. Okay. Reason he is mentioning like they are involved in what? DNA replication. Wrong. Okay. So assertion you look into carefully again. Ribosomes are called as protein factories of the cell. Ribosomes are never involved in DNA replication. Ribosomes, for example, rRNA is involved in the process of transcription and translational processes, but never in the replication. So yes, here A is true and R is false. In that case, what option we have to opt ma? Option 3 will be the right answer. Next question you look into, ribosomes will be composed of rRNA and proteins. How many different types of proteins will be there? 80 different types of proteins, and they exist freely or they are attached to endoplasmic reticulum in eukaryotic cell. So here what will be the right answer? Option B. And here once let us recall the positions or the sites of ribosomes in both prokaryotic cell and eukaryotic cell. In prokaryotic cell, if you have to look into, if I have to consider this as a cell, ribosomes, 70S ribosomes you can see both in cytoplasm and the ribosomes attached to cell membrane or plasma membrane. Eukaryotic cell, if you have to consider ma, eukaryotic cell, the ribosomes are identified in cytoplasm. Ribosomes are identified in cytoplasm and they are present adjoining to the endoplasmic reticulum. Ribosomes are present adjoining to the endoplasmic reticulum inside the cytoplasm, and they are present in the chloroplast and mitochondria also. In mitochondrial matrix ribosomes are present, and in chloroplast stroma also the ribosomes are present. Clear? So these are the 4 locations. In eukaryotic cell, where do you find ma? In cytosol, inside mitochondrial matrix, inside chloroplast stroma, and attaching to rough endoplasmic reticulum. And here you can see 80S, both of them will be 70S and this will be 80S. Clear, right? Next, this question you look into, which of the following organelles have their own DNA and 70S ribosomes? Which of the following will have their own DNA and 70S ribosomes? Their own DNA and 70S ribosomes are present in mitochondria and plastids. Okay, they are present where ma? In mitochondria and plastids. Option A will be the right answer. Okay, so that is why they are called as what? Semi-autonomous or self-duplicating organelles. Then you look into this question again, which is again an assertion reason type of question. So here if you see ma, in assertion it is clearly mentioned that eukaryotic cell or eukaryotic flagella exhibit 9+2 arrangement. Eukaryotic flagella will be exhibiting what? 9+2 arrangement of microtubules. Yes, right? So they are exhibiting 9+2 arrangement of microtubules. Correct. Prokaryotic flagella will show 9+0 arrangement. Eukaryotic flagella 9+2 arrangement. Once let us recall this microtubular arrangement of 9 plus 2 and 9 plus 0. So, here if you see 9 plus 2 arrangement, central 2 microtubules will be there and this central microtubules will be surrounded by a central sheath. Okay, this will be surrounded by what? A central sheath. From this central sheath, 9 radial spokes will be radiating out. Okay, 9 radial spokes will be radiating out and each radial spoke will have 2 peripheral microtubules. Okay, each radial spoke will be having how many microtubules 2 peripheral microtubules. So, this is 9 plus 0 arrangement. Whereas in prokaryotic flagella, at center proteinaceous hub will be there. This proteinaceous hub is having how many radial spokes? 9 radial spokes again. Each radial spoke will be having 3 peripheral microtubules. Each radial spoke is having how much? 3 peripheral microtubules. So, here it is having 2 peripheral microtubules. So, this is the arrangement of 9 plus 2 and this is 9 plus 0 arrangement. In reason it is clearly given the axoneme of cilia and flagella contains 9 peripheral doublets and 2 central singlets. What is axoneme here? The core of cilia and flagella is called as axoneme. If I have to take a flagella, here basal body region which is attached to the flagella body and hook and filament. These are the 3 parts of flagella. Okay. So, from this is said to be the core. Okay. If you take that core, that is called as what? Axoneme, which is showing this contains 9 peripheral doublets and 2 central singlets. Once let us again read assertion and reason. Eukaryotic flagella exhibit 9 plus 2 arrangement. Yes, that is true. And the axoneme of cilia and flagella contains how much? 9 peripheral doublets and 2 central singlets. So, in reason he is clearly explaining you about 9 plus 2 arrangement. In that case, what we have to choose? Option A. Both A and R are true and R is the right explanation of A. Very good, Tirupati, that answer is right. Continue answering the questions. We have 3 more questions. Okay. Match the following type of question over here. Centrioles. Just now we have discussed about centrioles, which has 3 peripheral triplets. Okay, centriole will show what type of arrangement? 9 plus 0 arrangement is shown by centrioles. So, here if you see, centrioles are having 9 triplets of microtubules. Axoneme is having what? 9 plus 2, 9 peripheral doublets and 2 central singlets. And a nuclear pore. Nuclear pore will regulate the transport between nucleus and cytoplasm. Next is what? Nucleolus. Nucleolus, which is present inside the nucleus, is helpful for the synthesis of rRNA, and inside the nucleoplasm tRNA and mRNA synthesis will happen. As such, if you have to look into options, option A will be right, where A is 1, B is 2, C is 3 and D is 4. Clear right now? Next question you see. Which of the following statements are true? So, here we have to identify the combination of the correct statements. Look into that carefully. Centrosomes organize spindle fibers during cell division in animal cells. Okay, this happens where? In the animal cells. B, if you see, nucleolus is a membrane bound organelle. Wrong. Nucleolus, which is present inside the nucleus, is not a membrane bound organelle. Then nuclear pores allow the bidirectional transport of RNA and proteins. Correct? Nuclear pore will allow the bidirectional transport. And then cilia and flagella are covered by plasma membrane. True. Cilia and flagella are covered by what? Plasma membrane. So, here what are the combinations of the right statements? A, C and D. Clear right ma? Next question you see. Which of the following organisms will have more than one nucleus per cell? So, look at here keenly. Option A is what? Chlamydomonas. We all know Chlamydomonas is a unicellular green algae and this is uninucleate. Okay, this is uninucleate. This is a unicellular green algae. Next, sieve tubes. Sieve tube cells are enucleated. Sieve tube cells are enucleated. Erythrocyte, which is mature RBC. Okay, erythrocyte, which is a mature RBC, this is also enucleated. paramecium which is given over here, this is binucleated, wherein paramecium will have one macronucleus and one micronucleus. So, this is having one macronucleus and one small micronucleus. Macronucleus and micronucleus. Macronucleus is aiding or helping for the process of what? Metabolic activities conducted inside the cell. Okay, metabolic activities. Whereas micronucleus will look into all the functions with respect to reproduction. It will control the reproductive process of the cell. Okay. So, here paramecium is a binucleated cell, it has macronucleus and micronucleus. Micronucleus will look after the reproduction, macronucleus to the metabolic activities of the cell. So, here what is the right option? Option C will be the right answer. Next question you see ma, here centriole microtubules are arranged in, centriole microtubules will be arranged in 9 plus 0, where it has 9 peripheral triplets, while axonemes will show 9 plus 2 arrangement. Okay, axonemes will show what? 9 plus 2 arrangement. So, here centriole is 9 plus 0, axoneme is 9 plus 2. In that case, what will be the right answer? Option B. Okay. Next, assertion and reason type of questions. Metacentric, this is about the shapes of chromosomes. Keep answering ma. This is about what? Shapes of the chromosome. Very good. Vishwant. Look at it once. Metacentric chromosomes has 2 equal arms. Yes, it has 2 equal arms and it will form V shape during the anaphase of cell division. In reason, it is showing like the centromere is located close to one end of the chromosome. Wrong, the centromere is located exactly at the center of the chromosome. Okay. So, here A is true and R is false. Option C will be the right answer. Okay. Next question. Yeah, this will be the last question ma. Just look into that carefully and then you can answer it easily. Metacentric chromosome, this has the centromere is at the center and it has 2 equal arms of the chromosome. This is metacentric. Okay, centromere is at the center. Option 4 will be the right answer. Next is submetacentric chromosome, where centromere is close to one end, is slightly away from the center. So, here option 2 will be the right answer. Acrocentric chromosome, centromere is near to one end and far from the other end. This is option 3. Next, telocentric. Centromere is at the terminal end of the chromosome. This is at the terminal end, which is option 1. So, here you look into the options carefully ma. A is 4, B will be 2, C is 3 and D is 1. Option A will be the right answer. Clear, right? So, with this, today's questionnaire session is done and we will come up with the chapter cell cycle and cell division tomorrow, where we will be exercising the most advanced questions of the chapter. So, stay tuned and thank you.